So today we are going to read about concept of pigment system. So let's start reading. In 1943, Robert Emerson and Charlton Lewis examined the action spectrum in the visible region for oxygen evolution in green algae known as Chlorella pyrenoidosa. For this green alga, these two scientists examined the action spectrum in the visible region for oxygen evolution. So by examining the action spectrum in visible region for oxygen evolution, they made this conclusion that the quantum yield remains fairly constant up to 680 nanometer, and beyond 680 nanometer this quantum yield declined sharply. This is the observation that they made. That is, quantum yield remains fairly constant up to 680 nanometer, but beyond 680 nanometer the quantum yield declined sharply. So this drop in the quantum yield in the far red region of the spectrum is known as red drop phenomenon. The far red region means the region beyond 680 nanometer. So in that far red region of the spectrum there is drop in the quantum yield, and this is known as your red drop phenomenon. So from red drop phenomenon we conclude that the light whose wavelength is greater than 680 nanometer is less efficient for photosynthesis and as compared to the light which have shorter wavelength. This simply means that if the light is having a wavelength greater than 680 nanometer, that is, it is in the far red region of the spectrum, then that light is less efficient for photosynthesis. And if your light is having the wavelength less than 680 nanometer, then that light is more efficient for photosynthesis as compared to this light. Now what is quantum yield? We are talking about it, and the quantum yield is the number of oxygen molecules which are produced per photon absorbed. The number of oxygen molecules which are produced per photon absorbed is known as quantum yield. When one photon is absorbed during photosynthesis, so because of that one photon which is absorbed, how many O2 are produced, this number of O2 produced because of one photon is absorbed, that number is quantum yield. When one photon is absorbed, how many O2 are produced, that number is quantum yield. The reciprocal of the quantum yield is quantum requirement. When you take the reciprocal of the quantum yield, that is 1 by quantum yield, then that is equal to quantum requirement. Quantum requirement means the number of photons needed for each oxygen molecule which is produced. Now how many photons are required to release one oxygen molecule? That number of photons which is required to release one oxygen molecule, that is your quantum requirement. In another experiment, Emerson and his colleagues set up an experiment with two beams of light. In this two beam of light, one beam had a red region, was emitting red region wavelengths, whose wavelength is less than 680 nanometer, and the other beam was emitting light in the far red region. That light had a wavelength greater than 680 nanometer. Okay, if the wavelength is less than 680 nanometer, then that is red region, and if the wavelength is greater than 680 nanometer, then that is far red region. And the two beams were emitting light in the red region, and the other beam was emitting light in the far red region. So Emerson applied two beams simultaneously. Okay, two beams were applied simultaneously, and it was observed that the rate of photosynthesis is 2-3 times greater than the sum of the rates which were obtained from each beam separately. Okay, so when you apply the two beams simultaneously, then the rate of photosynthesis is much greater as compared to individual beams. Okay, even if you add the sum of rate of photosynthesis obtained from each beam, then that sum is also much less as compared to the rate of photosynthesis which is there when two beams are applied simultaneously. That is, one beam in the red region and the other beam in the far red region. Both of them, when they are applied simultaneously, the rate of photosynthesis is much greater as compared to individual beams. Now, this phenomenon is known as Emerson enhancement effect. So let me explain you this Emerson enhancement effect in a simple way. Okay, consider a tube. In this tube there is a suspension of photosynthetic organism. Okay, now this tube is exposed to wavelength which is greater than 680 nanometer and the wavelength which is less than 680 nanometer simultaneously. Okay, these two wavelengths are hitting the tube simultaneously, and they are causing a particular rate of photosynthesis. Now, in the second condition, what is happening is that this same tube we take and we hit this tube with a light whose wavelength is greater than 680 nanometer, and after that we hit that same tube again with a wavelength that is lesser than 680 nanometer. Okay, these two wavelengths are not hitting the tube simultaneously, they are hitting the tube separately, and whatever the rate of photosynthesis we obtain from these two separate, we just add them. Okay, and we get a particular rate of photosynthesis. And in this case, when the two wavelengths are hitting the tube simultaneously, we get a particular rate of photosynthesis as well. Now, this rate of photosynthesis is 2 to 3 times greater than this rate of photosynthesis, and this is the Emerson enhancement effect. That is, these two wavelengths, when they are hit simultaneously, are causing more effective photosynthesis as compared when these two wavelengths are acting separately on the same tube. Okay, this is your Emerson enhancement effect. Now, this enhancement effect that we just saw implies that the photosynthesis involves two photosystems. One photosystem is driven by a longer wavelength, that is greater than 680 nanometer, and the other photosystem is driven by a short wavelength that is less than 680 nanometer. Okay, there are two photosystems. One photosystem is driven by a longer wavelength and the other photosystem is driven by a shorter wavelength. So this graph is representing the Emerson enhancement effect. Okay, here you have rate of photosynthesis, and so when you keep a light of you are hitting the tube with a light which has wavelength greater than 680 nanometers, so this is the rate of photosynthesis you get. Then what you do is you hit that same tube with a light whose wavelength is greater than 680 nanometer, and after that also you get this much amount of rate of photosynthesis. But when you hit the same tube with the wavelength that is greater than 680 nanometer and which is less, and also with the wavelength less than 680 nanometer, you hit the tube with the two wavelengths simultaneously, then this is the rate of photosynthesis you get. And as you can see, this is much greater than the addition of these two, and this is your Emerson enhancement effect. So the Emerson enhancement effect is that the rate of photosynthesis when the red and far red light are given together, this rate of photosynthesis is far greater than the sum of the rate of photosynthesis which is obtained by giving this red and far red light apart or giving them separately. This is Emerson enhancement effect. Now this enhancement effect provided essential evidence for the theory that the photosynthesis is carried out by two different photosystems, and these two different photosystems work in series and they have slightly different wavelength optima. Okay, these two different photosystems have slightly different wavelength optima, and these two different photosystems work in series, and the photosynthesis is carried out by two different photosystems. This hypothesis, the essential evidence for this hypothesis is provided by the enhancement effect. Now let's discuss about pigment systems. In all natural photosynthetic systems, the pigment molecule is bound with protein. Okay, and when the pigment molecule is bound with protein, that complex is known as pigment protein complex, simple. And this pigment protein complex is also known as the pigment system or photosystem. So your pigment system or photosystem is a pigment protein complex which is formed when the pigment molecules are bound with proteins. Now this pigment system that we are talking about is made up of two components. Okay, the first component is photochemical reaction center, and the second component that makes up your pigment system is antenna complex. Photochemical reaction center and antenna complex are the two components of pigment system. So photochemical reaction center is a protein pigment complex that carries out the photochemical reactions. Okay, it is a protein pigment complex that carries out the photochemical reaction. So in your reaction center, the photochemical reactions are taking place. So the photochemical reaction centers are also of two types. FES type reaction center, which is known as type 1, and pheophytin quinone type reaction center, which is type 2. This type 1 and type 2 are the two types of the photochemical reaction centers. Okay, FES type and pheophytin quinone type. Now, antenna complex is also known as light harvesting complex, and it is made up of a number of distinct protein pigment complexes. Okay, the antenna complexes is made up of many pigment protein complexes. Now this antenna complex is made up of two components. Okay, the core antenna, or which is also known as inner antenna, is the first component, and the second component of antenna complex is peripheral antenna or outer antenna. Okay, so core antenna and peripheral antenna are the two components of the antenna complex. This core antenna is also known as inner antenna, and this peripheral antenna is also known as outer antenna. Now, what antenna complex does? Antenna complex captures the light energy, and then antenna complex feeds this light energy to the reaction center. Okay, the light energy from the antenna complex is supplied to the reaction center by a process known as resonance energy transfer. Now the antenna complex is able to feed the light energy to the reaction center through a process known as resonance energy transfer. So this is how it looks. The light energy is captured by the antenna complex, and that energy is then transferred to the reaction center by a process known as resonance energy transfer. Now the size of this antenna complex vary within different organisms. Okay, some organisms have large antenna complexes, some organisms may have small antenna complex. It varies with different organism, and it varies considerably. Now generally the higher plants have 200 to 300 chlorophyll molecules in the antenna complex. Higher plants have 200 to 300 chlorophyll molecules in the antenna complex. That is, it generally happens that. So all photosynthetic organisms have the antenna complex, but Heliobacter is an exception. Okay, Heliobacter is a photosynthetic organism that do not contain antenna complex. Okay, except this Heliobacter, all the photosynthetic organisms contain antenna complexes. So this is how your antenna complex and the reaction center looks like. Okay, so the light energy comes and it is accepted by one of the chlorophyll of the antenna complex, and then that light energy is transferred to other chlorophyll molecule, and then to other, and then to other, and then to other, and finally it reaches the chlorophyll of the reaction center. Now here that energy, like the high energy electron is transferred to an electron acceptor, and low energy electron is accepted from a donor, and that is how the chlorophyll in this reaction center comes to the ground state. That is how the energy flow occurs between the antenna complex and the reaction center. So this diagram is showing the organization of the pigment system in the thylakoid membrane. So the pigment system which we just saw in the diagram is tightly packed in the thylakoid membrane. Okay, your thylakoid membrane contains this tightly packed pigment system, and the pigment system contains several hundred of antenna complexes and accessory pigments which are surrounding the photoreaction center. Okay, so the photoreaction center is in the middle, and around this photoreaction center there are several hundreds of antenna complexes and several hundreds of accessory pigments, and this is how your pigmented system exists in thylakoid membrane. Now when there is absorption of photon by antenna complex, then this leads to the excitation of the reaction center, and this happens because of excitation transfer. Okay, the energy is absorbed by the antenna complex, that energy is transferred to the reaction center. So when the photochemical reaction happens in the reaction center, that photochemical reaction converts the photon energy into the separation of charge, and this separation of charge initiates the electron flow. Okay, so the photon energy is used to initiate the electron flow. That is your photochemical reaction which takes place in the reaction center. So by photochemical reaction you are using this photon energy to initiate the electron flow. So this is how the overall picture looks like. The light energy is absorbed by the antenna complex, and that energy is transferred to the reaction center. Here the photochemical reaction takes place and there is initiation of flow of electrons. So this light energy is utilized to initiate the flow of electron. That is how it takes place. The overall picture is. Now in oxygenic photosynthetic organisms, that is plants and cyanobacteria, both type of photosystems are present. Okay, the one which drives the longer wavelength and the one that drives the short wavelength, those both kind of photosystems are present in oxygenic photosynthetic bacteria, that is plant and cyanobacteria. So we saw this, right? The photosystems are of two types, photosystem 1 and photosystem 2. The photosystem 1 has the FES type of the reaction center, and the photosystem 2 has PO15 QE type of the reaction center. So this both photosystem 1 and photosystem 2 are present in the oxygenic photosynthetic organisms such as plants and cyanobacteria. So photosystem 1, let's look about in detail about photosystem 1. So photosystem 1 is abbreviated as PS1. It is a protein pigment complex that uses light energy to mediate the electron transfer from plastocyanin to ferredoxin. Okay, so in photosystem 1 there is electron transfer from plastocyanin to ferredoxin, and this electron transfer is mediated with the help of the light energy. So photosystem 1 uses the light energy to transfer the electron from plastocyanin to ferredoxin. Now this photosystem 1 has the FES type of the reaction center. This photosystem 1 contains protein molecules, chlorophyll A molecules, carotenoids, phyloquinones, FES center and other cofactors. Okay, protein molecules, chlorophyll A molecule, carotenoid, phyloquinones, FES centers and other factors are present in your photosystem 1. The cyanobacteria contains a photosystem which has 11 proteins. Okay, the photosystem 1 of the cyanobacteria has 11 proteins. They are PSA A, PSA B, then PSA C, D, E, F, I, J, K, L, and then PSA M. Okay, these 11 proteins are present in the photosystem 1 of the cyanobacteria. Now the photosystem 1 is driven by the light of wavelength 700 nanometer. Okay, Photosystem 1 is driven by the light of 700 nanometer. Photosystem 1 700 nanometer. Now there is chlorophyll A molecule present in the photochemical reaction center of PS1, photosystem 1. In photosystem 1 there is a photochemical reaction center. At that photochemical reaction center you have your chlorophyll A molecule. Now that chlorophyll A molecule is known as P700. Now why this chlorophyll A molecule is known as P700? Because it most effectively absorbs the light of wavelength 700 nanometer. Okay. The chlorophyll A molecule, which is present in the photosystem 1, which is present in the photochemical reaction center of the photosystem 1, this chlorophyll A molecule most effectively absorbs the light of wavelength 700 nanometer, and that is the reason why this chlorophyll A molecule is known as P700. In this name, that is P700, the P stands for pigment. So pigment 700 means the chlorophyll A present in the photochemical reaction center of photosystem 1, and this chlorophyll A is known as pigment 700 because it most effectively absorbs the light of wavelength 700 nanometer. That is why pigment 700. Now let's talk about photosystem 2. Let's read about it. Now this photosystem 2 is abbreviated as PS2, and it consists of pigment protein complexes, and it also consists some proteins and cofactors. Okay, photosystem 2 is a pigment protein complex which contains several proteins and several cofactors. Photosystem 2 contains a reaction center of pheophytene QO9 type. Okay, pheophytene QO9 type of reaction center is present in your photosystem 2. Now the chlorophyll A, which is present at the reaction center of the photosystem 2, is known as P680. Okay, the photosystem 2 has a reaction center. That reaction center has a chlorophyll A molecule. That chlorophyll A molecule is known as P680. Why? Because this chlorophyll A molecule most effectively absorbs the light of wavelength 680 nanometer. Okay, this chlorophyll A molecule most effectively absorbs the light of wavelength 680 nanometer, and that is the reason why this chlorophyll A molecule, which is present at the reaction center of photosystem 2, is known as P680. So photosystem 1 has a P700 and photosystem 2 has P680. Now these two pigments, that is P700 and P680, are identical chlorophyll A molecules, but the fact is that the P700 absorbs show maximum absorption at 700 nanometer, and P680 shows maximum absorption at 680 nanometer, but both of them are identical chlorophyll A molecules. The thing is that there is the P700 and P680, these two pigments associate with different proteins, and this thing account for their slight differences in light absorbing properties. Okay, P700 is associated with different proteins, P680 is associated with different proteins, and that is the reason why the P7 shows maximum absorption at 700 nanometer wavelength and P680 shows the maximum absorption at 680 nanometer wavelength. Okay, because of their association with different proteins, they account slight differences in their light absorbing properties. So in higher plants, the reaction center contains membrane proteins such as D1 protein, D2 protein, and cytochrome B599, and it along with that it also contain other proteins, and the reaction center also has chlorophyll A molecule, pheophytin, plastoquinons, and other cofactors. So this is the composition of the reaction center in higher plants. Okay, membrane proteins such as T1, D2, and cytochrome B599, other proteins, then chlorophyll A, pheophytin, plastoquinons, and other cofactors. Now the reaction center is associated with light harvesting antenna complexes, and we know that light harvesting antenna complexes are of two types, that is core antenna and peripheral antenna. Now let's talk about distribution of photosystem 1 and photosystem 2. These two photosystems, that is PS1 and PS2, are distributed differently in the thylakoid membrane of the chloroplast. So your chloroplast contains thylakoid membrane, okay, and in that thylakoid membrane the two photosystems are distributed differently. Okay, there is no homogeneous distribution. The distribution of the two photosystems is quite different. That's the point. Now PS1 is found exclusively in the non-appressed membranes of the grana thylakoid and stroma thylakoid. You have your grana thylakoid and stroma thylakoid in the non-appressed membranes of grana thylakoid as well as stroma thylakoid. The PS1, the photosystem 1, is found exclusively. The PS2, the photosystem 2, is found predominantly in the appressed membrane of the grana thylakoid. Okay, grana thylakoid have appressed membrane. In that appressed membrane you have PS2. So yeah, grana thylakoid have non-appressed membrane as well as appressed membrane. Now in the non-appressed membrane of the thylakoid you have your photosystem 1, that is PS1, and in the appressed membrane of grana thylakoid you have predominantly photosystem 2, that is PS2. So this diagram clarifies it. Now this is your grana thylakoid, this is your stroma thylakoid. Now the green structure that you see is non-appressed membrane. Okay, that is, it has a free surface, it is non-appressed membrane, and the white is appressed membrane. That is, this membrane is not free, is kind of appressed upon each other. So in this non-appressed membrane, which is free, photosystem 1 is found, and in the photosystem 2 is found in this appressed membrane of the grana thylakoid, and PS1 is found in the non-appressed membrane of the grana thylakoid as well as stroma thylakoid. Okay, stroma thylakoid simply don't have appressed membrane. Okay, only grana thylakoid have the appressed and non-appressed one. Now the outer membrane surface of the thylakoid is divided into two structurally different regions. So this outer membrane surface of the thylakoid is divided into two structures, that is appressed membrane and non-appressed membranes. So the appressed membranes are found in the grana thylakoids. These appressed membranes are contact with other membranes. Okay, and then there are these non-appressed membranes which are found in the stroma thylakoids and which are also found at the ends and margins of grana thylakoids. These non-appressed membranes are exposed to stroma. So this non-appressed membranes contains PS1, photosystem 1, and this appressed membrane contains PS2. Okay, so PS2 is kind of in contact with other membranes and PS1 is exposed to stroma. So thanks for reading with Muffin. That's all for this video.